a little. And also I'm a researcher at Institute DOR and visiting researcher at University of Ceará. That was the place that I did my bachelor. I also was a co-editor in that book, the Handbook of Low-Level Laser Therapy. That is one of the important book in photobiomodulation, that is the use of light to interact with cells and change the way to modulate that cells, and basically use that for therapy, majorly pain, inflammatory, and regenerative medicine. So by now, I separate that presentation in 5 parts. Don't worry, it is very quick. It's just to keep everything in mind. So the first part, a little bit about the basics of quantum mechanics, to explain for a broader attendance about duality and probability in wave functions. The second part is more connected with the chapter of the book by itself. So we talk about quantum photosynthesis, the physiology and the light harvesting, and the quantum beating energy transfer. So that's the how can I say, the most important part, or actually the most connected part with the book, the John Joe's book. But also I will very quickly talk about another 2 effects of photobiology, photomedicine, that is the energy transduction in vision, in the bastonets in your retina, and also about quantum effects in photobiomodulation. That is the science that I worked during my PhD, and also is the science that we work in Bright. Just to finalize, we will very quickly talk about photosyntheticals, that is technology to personalize photobiomodulation. But I am sure that last 3 parts is very quick. So the part 1 and part 2 are the ones that we will focus most. So the thing is, classical mechanics and quantum mechanics are very different in concept. For example, in classical mechanics, we have, if we know the forces that are interaction, we can know the position, velocity, acceleration of the objects. And here I put the trajectory of Voyager 1 and Voyager 2, and we can know very precisely where space probes are, because we know the forces that are interacting with them. However, when we talk about quantum mechanics, we have only access to the wave function that allows us to know a little bit of the probability distribution, not the position or velocity by itself. While photons are particles, they behave like particles and like waves. In the 19th century, the photons, the light, and actually were proved to be a type of electromagnetic radiation, and that electromagnetic radiation behaves like waves. But in the 1905, Einstein explained the photoelectric effects considering the quanta of light, the photons, as a particle that can interact and take off electrons from the metal surface. So showing that in specific cases, the light can behave like a particle that was named the photons. Also very interesting is that particles, like for example, electrons, they can interact and interfere with themselves like waves. So showing the interference pattern, and it was proposed by Debris. And after that, Schrödinger proposed an equation that has a wave function as the entity that we physicists can work with, that psi, that is the distribution of probability. And that explanation, that interpretation, connecting the wave function with probability, was made by Max Born, and the square of psi was interpreted as the probability to find the particle in that specific position, in that specific time. So when we talk about quantum mechanics, the best information that we can have is that distribution of probability, and not the position, velocity, acceleration, as we had in classical mechanics. Of course, I'm talking broadly, I'm talking to a broad audience, and of course we can have more, can be deeper in that explanation, but in general, we can understand that. And just to present a few differences about quantum mechanics, classical mechanics, and quantum mechanics. For example, if we have an harmonic oscillator, in classical mechanics, we can know position, velocity, acceleration, and also it can have a continuum of energies. In quantum mechanics, an oscillator like that, an harmonic oscillator, we only can know the wave functions, the distributions of a very specific, very discrete energy levels. So here we can see an interesting difference. As we told before, in classical mechanics, we have deterministic and precision, and in quantum mechanics, we have probabilities and we have discrete energy levels. Extrapolating, when we talk about atoms, molecules, we have the same pattern, that we have wave distributions, wave functions that are probability densities, and also energy levels that we can find in the systems. Here, the system is the hydrogen atom, that is the easiest system possible for an atom. And so I'm just saying here that when we see probability distributions and also energy levels, we are talking, it's a signature for quantum mechanical effects. Now, entering that second part about the quantum photosynthesis, and I will present to you how it happens, the photosynthesis, and in the second moment, how quantum mechanics can be correlated to that. So we can see it is made by cells, of course, and inside one of that cells, we have the chloroplasts, and that are an organelle that is responsible to make photosynthesis and transform basically the gases, that the air and the water plus photons, and create the matter, the life matter of the plant. The carbons come from the air and are transformed in substances of the tree, of the plants, due to the photosynthesis that happens in that chloroplasts. And inside the chloroplasts, we can see the thylakoid, that is that little discs here, and is the real place where the photosynthesis happens. And the light reaction takes place on the thylakoid membrane to generate ATP, and due to the Calvin cycle, it takes off takes light, water, and carbon dioxide, and in the Calvin cycle, it is transformed in substances to the plant, organic molecules that are used to build up the plants, build up the trees. So it's just a parenthesis. In the beginning of the chapter 4 of the Life in the Edge, they present a text by Richard Feynman that is very poetic text, saying that basically we always are used to think that the tree brings up the matter from the ground, but actually the matter that build up the plant comes from the air, and together with water and light, it becomes the plant itself, the molecules, the organic molecules of the plants. So the place that it happens is in the thylakoid, in the membrane of the thylakoid. That is the place that Calvin cycle happens. So using water, carbon dioxide, and light to provide that, consuming ATP to create that organic molecules. So the thing is, we have several antennas that capture the photons, and that photon can extract, when it is absorbed, it makes an electron goes away from that structure of that antenna. And so it becomes an unconnected electron, an electron that is not connected to any of the molecules, and a hole. These both together, the electron and the hole, are known as excitons, and it contains the energy. It is the energy that came with the light, with the photons, are armazenated some way in that excitons. The thing is, there is only one reaction center for several of that antennas. And the people didn't know how the type of energy transfer could be so efficient. The efficiency is really, really high. Actually, is probably the most efficient transfer of energy known, naturally or artificial. And if we have very precise conditions, it is approximately 100% efficient. It means all the energy that comes in the photons are really transformed in chemical energy of the plant. But when classical mechanic and electromagnetic explanations could not fit the data, they could not fit that so precise and so big efficiency, because one of the hypotheses was that that excitons could go jumping randomly from one antenna to the other until to find the reaction center. Of course, many of that exciton, that is a very reactive entity, would be lost in the way. In femtoseconds, it would be lost. So it could not be explained only by the electromagnetic and classical mechanics. So a group working in California found some evidences for wave-like energy transfer through quantum coherence in photosynthetic systems. What it means is that different from a localized exciton as a particle here jumping from one place to the other, what they were saying is that that exciton have a wave function that would be distributed over several of that antenna and over the reaction center. So it would always know where the reaction center is. And to prove that, to find that evidence, they use a technique named as two-dimensional Fourier transform spectroscopy. As the name says, spectroscopy, we can find several energy levels. It's a type of spectroscopy. And Fourier transform is a technique that can change position and frequency to can change frequency by time or position to the other type of wave-like number. And the thing is that they found here the distribution and oscillation, that is a type of signature of quantum mechanics, as we told before, and found energy levels of transition. And if it was only classical, it could have a broader peak of energy, a continuum of different energy, and would not have that type of peaks here. So that's broadly speaking, a signature of quantum mechanical effects, like for example that data here showing the intensity over the population in femtoseconds, and that type of oscillation here is a signature of quantum effects. If it was only classical, we could have not oscillation but a continuum of points, or only a peak of absorption, but not an oscillation like that. So somehow that is a signature of quantum mechanical effects. So that's very interesting. And also I bring here some, how can I say, possibilities of quantum effects in different, not only in plants, but also in animals. Here, I will very quickly talk about the possibility of quantum effects in the retina. Now, for simplicity, I will talk only about the bastonets that are the ones, the type of cells that absorb lights when we are in a very dim condition. So here is the way that they are, and here the outer segment, inner segment, the place where the absorption happens, the photon absorption happens here in that outer segment, and when the photons are absorbed, it despolarizes that outer membrane that makes to the next cell that is connected with the bastonet will be despolarized too and create action potential. So that actual potentials that happens when there is an photon absorption. Millions and millions of them each second are the ones that provide us to create the sense of vision. Here is to show that that type of membranes where the absorption happens. And in that membranes, there is a molecule that have two possible conformations, that is the retinal. And when light is absorbed, there is a change process retinal to transretinal that change the conformation. And that change of conformation is the thing that makes the the change in energy levels, that is the one that can create the depolarization of the neuron, the rod neuron. So the thing is, if we have only one transformation like that, it would not be possible to create the action potential in the next neuron. And the thing is that it's in the last years are being mounted evidence that to really create the action potential, it would be interesting, it would be more effective for that rods to have any type of connection, long distance connection, that would be necessary, some type of coherence to the absorption of more than one photon simultaneously to create that action potential. And it's one thing that our group in IDOR is studying right now, that is the possibilities of absorption of photons that are emitted for quantum optic sources, and if it has differences from traditional classical optical sources, and if it would have any type of difference, not only in the electrical retinal signals, but even in more and deeper brain regions using magnetic resonances. As I told, it's a very, very interesting research, but we just started it, and the first experiments will be done in the next year. Just to finalize the presentation, I would very quickly talk about photobiomodulation, because it's the area that I studied, and because right now is being mounted some events for quantum mechanics, quantum biology effects in photobiomodulation. Photobiomodulation happens here with visible and near infrared light. It's not thermal, like far infrared, and also it's not connected for damaged DNA as ultraviolet light, but it is connected to stimulate the mitochondria, precisely the cytochrome C oxidase, to enhance the production of ATP. And with more ATP, the cells are able to produce more substances for endogenous analgesics or anti-inflammatories or growth factors. So the basic science of photobiomodulation is provide photons. It increases the rotatory motors that are used to produce more ATP, and with more ATP, the cells can work by themselves to produce more of the substance that they need most in that moment. And right now, the first dogma of photobiomodulation was that somehow the photons would be absorbed by cytochrome C oxidase, as I told, and we didn't know why it would improve the velocity that molecular motor works. And what that very interesting study here presented is that it can increase, it can decrease the, it would be a fuel, but also a lubricant for the water in the, that is near the rotatory motors. So it could decrease the fluidity of that water and due to a very quick transient effects in order of femtoseconds of and it is being studied by that group too, and also has Marlan Scully, that is a big name in quantum optics, studying that type of effects in cytochrome C oxidase in the regions near it. And also, as a curiosity, the quantum dot, excuse me, actually quantum dots are being studied as emitters for, as sources for photobiomodulation too, and photomedicine in general. Well, just to finalize the presentation, I will talk about photoceuticals. That is the possibility to use photobiomodulation in a personalized way that allows the physicians to prescribe the specific amount of light to be absorbed in a very specific part of the body to induce the body to produce the very specific substances. As I told, analgesics, endogenous analgesics or anti-inflammatories or growth factors. So it would be somehow the use of photons as today it is used, the drugs, the pharmaceuticals, so the name photoceuticals. I started to study that during my time in Harvard Medical School, 2013, and we made the preclinical studies connecting that absorption of light with the modulation of very specific substances. So we could find, for example, correlations of nanojoules per cubic millimeters to produce specific amounts of nanograms of specific substances. And it was very interesting. I think that I received, my thesis received one of the most important prizes in Brazil for thesis, most because of that findings. And I believe that the invitation to write the handbook of photobiomodulation was due to that study here. I will not wait too much in the details, but basically we made preclinical study with mice, and we could find analgesic effect connected with pain mechanism related to heat, cold, inflammation, pressure. So several behavior tests in animals, and we could find the precise time scale that what was happening. For example, after the photobiomodulation, we saw the ATP increases. After that, the analgesic PAP here increases. After that, the decrease in glutamate, that is a neurotransmitter connected to the pain signaling. And here, just to show the behavior of the animal, we have the control group that do not change the behavior after the treatment. Here, 0 is the baseline, and a group that received the precise amount of photons that we saw an increase in 3 times the tolerance of the animal to the pain stimulus. So right now, I will not enter in so many details, but the company that I founded, Bright Photomedicine, it works is precisely in the creation of that photoceuticals that, as I told you, are the stimulus with photons to enhance the production of endogenous substances, endogenous remedies. Of course, it must be personalized. Otherwise, it would not be possible to provide the same amount of light in the specific place to provide the therapy. So it is necessary a huge effort in computational physics to personalize that emissions. And right now we did it around 14,000 times that types of photosyntheticals were emitted, and we have clearance here in Brazil, and in the next few years we will seek for clearance in United States too, and Europe. So guys, and I really believe that quantum biology will be connected with the next steps of photosyntheticals to increase the predictability of what photons will, how the photons will be absorbed, to increase the previsibility of the type of substances that will be produced. So that was the reason that I connected with the quantum biology, the quantum bit. So that was what I prepared. Thank you very much for your attention. I think we have time for questions if you need, if you want, of course.
Yes, thank you very much, Marcelo. And if you want, you can just unmute yourself and ask all the questions that you want. Otherwise, just drop them in the chat and we will read them out for you. Okay, so Andrew is asking, how large is the pharmaceutical market from your perspective and expected time to market?
Thanks so much for the time.
You're muted.
Sorry, I muted myself and I forgot. Okay, actually, the pharmaceutical market is as big as the pharmaceutical market in some way, because it can technically almost any type of substance that have therapeutic effects can be produced by our own body. So basically, in thesis, in principle, it could be possible to enhance the production of our own body of analgesics, anti-inflammatories, regenerative medicine, neuromodulation, modulation of the autoimmune system. So it is very in the beginning to say, but the perspective, the total addressable market is $1 trillion total addressable market. So it's similar to the size of drug market.
Are you able to commercialize it the same way? In that, like, typically, you know, hearing stuff like this, are you able to sort of extract value from end users or patients in the same way as you could with pharmaceutical market?
Yes, actually, it's the way that we are doing in Brazil right now. We have clearance, and also we are marketing it as it was a drug. For example, the physicians prescribe today. They are prescribing. Actually, the company is in the very beginning, but we are very excited. Laura asked about the applications in inflammatory modulation. And yes, of course, actually, photobombutilation started. One of the biggest applications are in inflammations, because it can be used to decrease the swelling. And actually, it is possible to use in several types of inflammation, not only the musculoinflammatory inflammations, that is the ones that comes first to our mind, but for example, some types of dementia, they create inflammatory signals in the central nervous system, and that type of application transcranially can not only modulate, doing neuromodulation like electrical or magnetic modulation, but also can work in the decrease of that type of inflammation that are somehow related to deleterious effects of dementia, Parkinson, Alzheimer's, etc. So the thing is that our body is all the time producing inflammatory and anti-inflammatory markers, and there are even drugs sometimes are connected to the stimulus to enhance the body production of that anti-inflammatory or pro-inflammatory. And of course, there are drugs that are per se anti-inflammatories. So the idea is that it was proven that the increase in ATP in cells that are damaged, it is related to the production of that cells, enhance the production of interleukins, for example, interleukin 6, interleukin 10, that are very well known anti-inflammatory substances. For very interesting, Laura. She was saying that is exactly studying neuroinflammation and in other situations that are not so traditional, because every time that we thought about inflammation, the usual is thinking about musculoskeletal inflammation. But of course, inflammation is a reaction of the body that is in several types of tissues, not only in muscles.
Yeah, may I say something? I'm
Please.
Laura. It's absolutely interesting, Marcelo. Thanks. And we are working with patients, and we are applying electromagnetic field in patients with problems at neurological level. So we are seeing some beneficial effect. And I'm pretty sure that quantum effect is underlying what we see. The problem is that we are trying to create the experimental model, appropriate experimental model, and we are working on it. And also we have problem with people suffering obstructive sleep apnea and hypertension. And the mechanism underlying is like a vicious circle of neuroinflammatory patterns. And the idea, this is emerging, is that perhaps quantum, the quantum behavioral cell might be at the base of this network, because it's a very incredible network.
Exactly. Laura, it's very interesting. I would like to only talk, to do not run so away from our discussion here, but what I would like to say about the connection of photobiomodulation with quantum effects is the thing. The cells, of course, they must communicate with each other. And if we think about evolution, if you have electrical connection that in embryonary stages is the most common, the electrical signalings in cells that are closed together, but also there is the communication of cells using substances, chemical substances, but it's not very good in the sense that the cells cannot communicate well, in the sense that a substance can create several effects, and a type of communication between cells that are enhancing the research about it in the last decade is the photon, the communication, cell-to-cell communications using photons. So in some way, that would be a type of quantum effects for the information information exchanges in organisms. So it would be somehow connected with the photobiomodulation.
Excuse me, I think I missed I missed a word for a bad connection. Which is the type of information? Quorum? I missed the word.
Okay. Actually, it would be the exchange of information using photons.
Yes. Photons.
Yes,
Yeah.
exactly.
Photons. Yes.
Exactly.
Yes. Okay. Thanks.
So there are several papers about photonic communication, cell-to-cell photon communication of photons absorptions. So it's a very interesting new field. The thing is, talking about photobiomodulation or talking about photobiology in general, we do not have the sensing methods adequate to find the quantum effects. When we irradiate a cell, it's like a storm of photons. And what would be interesting is to see what one photon being absorbed in a very specific place, what happens. You
was
know,
the problem that block our experiments exactly.
Yeah. I don't know, and to be completely sincere, I really don't know how to overcome that issue, but it would be very, very cool. For example, there is people like Marlon Scully that I showed one of his papers that is studying femtosecond phenomena. I mean very quick phenomena, and of course in very specific targets. People in the discovery or the explanation of that atomic level, molecular level phenomena to the application of that knowledge to create some pharmaceuticals or some improvement in pharmaceuticals. It would be more few years or some years of research. But of course, that field is very interesting, and it will be a hot topic for research in the next years, for sure.
Okay, thanks you so much. Really remarkable. Thanks.
Thank you.
Okay, there are still many questions for you. So one is from Agamrup. I hope I'm pronouncing the name right. How do you see the photosynthetical research being done into the future? And do you see the research being applied to disease and other therapies as well?
Yes, for sure. Actually, I will do parallel. I will make a parallel with for example, the pharmaceutical companies. Actually, to do drug discovery right now, it's it's a mess. It's terrible because from the molecules, the principles to the approval of the FDA, it takes somehow an average 10 to 15 years. and $1 to $2 billion. So it's a huge amount of money, and it takes too long. So the thing is that even the big pharmaceutical companies, they are trying new stuff to overcome that financial crisis in the drug discovery, in the therapeutic discovery. For example, GlaxoSmithKline, GSK, invested a huge amount of money in what they call it electroceuticals. That was basically electrical stimulus directly in the peripheral neurons to change the signaling of that neurons and try to solve different diseases that goes from chronic pain up to muscle spasms or muscles weakness, and they had very good results, and it becomes a new field. So the photosyntheticals is connected, just making a parallel with that. They are being, time after time, being improved and becoming a scientific research field by itself. And I really believe that few years from now, 5 to 10 years from now, we will have universities and research centers working on that, in the same way that we have drug discovery being done in universities, in academia, and the improvement of medical devices being proposed by engineers, not only in companies, but also in universities. So I really think that the research, that it will be a research field by itself. Mm-hmm.
And we have another.
I have Aganrup, if I have not been clear, please let me know, and I try to explain another way.
Okay, and then we have another.
Sorry.
Oh, please.
I was just gonna say that was really clear. I was just wondering, you mentioned earlier about devices and like not being able to measure the quantum effects clearly. Do you think that research would first need to go into actually making those devices before we go further into like disease management and things?
Mm-hmm. No, actually, I don't see the things in that way, but it's a very personal way to see the things. I really believe that this both goes together. I mean, it is possible to make photosyntheticals even without knowing the quantum effects going on in a very specific molecules, in the same way that pharmaceutical companies and universities can create some drugs that they do not know perfectly what is going on in biochemical reactions, but they can see the therapeutic effects. But in the other hand, I see that as much as we know the basic science and why the things happens, we will be better prepared for predictability. We will be better prepared to overcome some issues and to discover more precise ways to induce the body to produce exactly the substance that we want. So I really believe that it will be a transdisciplinary area, because we must know a lot of the physics, but also biochemistry, and also computational simulations and big data analysis to be able to go deeper and deeper and be as much as possible, as precise and as predictable as possible.
Thank you so much.
Then we have a question from Kevin, which is asking what type of magnetic sensor is used in your experiments, I guess.
Okay. Actually, right now, we do not. Probably he is talking about the experiments in the retina to find quantum effects in the retina. And we are using not necessarily a magnetic sensor, but a camera that is capable to capture very dim light. Actually, units of photons can be captured by that camera. So this is the way that we are trying to produce by the same quantum optic source of photons, produce two entangled photons and send one to the camera and the other to the eye to see what happens when it is absorbed in the retina, or in the contrary, when it's absorbed by the camera. But as I told you today, it is more, how can I say, it is very theoretical, because we are the setup for the experiments is being prepared right now. But the idea is that next year it will be a platform for several experiments, not only in the retina, but also, for example, for the emission and absorption of photons from the living cells of animals and even plants. So and as we can do spectroscopy of very, very dim light, as I told, 1 by 1 photons, it would be interesting to see if there is a pattern that could be interpreted as the communication of cells, cell by cell communication. So we are very excited with that, to be clear.
Now, is that quantum ghost imaging that you're talking about there?
Exactly. Perfect. One of our co-workers, colleagues in that research is Gabriela Lemos, that is a big name, not only in Brazil, but in the world, about the ghost images. And the idea is, one of the ideas is that, is it possible to capture ghost images in your retina? And of course, if it would be possible, what would be the evolutionary necessity of that? Because we do not see that type of image from the sun, it's a very artificial type of image. So
there were some good papers more recently in ghost imaging. I think this year, subshot noise. I don't know if you do that, like quantum subshot noise. So basically all this stuff is to break the diffraction limit, Rayleigh, those types of things as well. And in specific, the one that's moving out as far as quantum imaging is quantum sensing, and these quantum diamond sensors. And they've been used from anything from a rat's heart to removing noise, but the technique called QPSD, which might be implicated in a lot of things in medical. They have another one, kind of spin amplification, that reduces readout noise too. So there's a lot of good stuff surrounding this technology that started in about 2009, when NIST said that you could put it on the tips of probes, like an MRI scanner, but just a lot smaller. So they figured out the algorithm last year, and we're talking about fluorescence. And then this year, in August, those two big ones with the rat's heart and nature, and then also quantum phase, something SD. and basically taking an MLK audio clip, removing noise. So that one and the spin amplification one, and also the MIT, there's an MIT article, it's all frequencies they can detect with these quantum sensors. And, if you type in, all frequencies MIT, you can see that one too. But you're going to see a lot of that incorporated in all sorts of even neuroimaging and other parts of medicine too. It's in the early kind of, early application, like they just hit the Nature papers. There's an EV battery one too as well, going to increase certainty from like 10% to within 1%, basically with charge. And then, so imagine that with non-invasive medical devices.
Yes, great. Thank you for letting me know. In that part of that experiment, I am working much more as a coordinator of the scientists than the principal investigator of that. So, of course, I will ask for the people that is more specialized in that in this experiment. I will bring your informations to them, and of course, it will probably be very useful for them.
Yeah, and of course, so I put the Google one in the chat. So that one's really important. It's quantum machine learning. So you need a quantum computer, quantum sensing, quantum algorithms for those. And then, so the issue with the computers is the noise, but using this layout with QML, they say that's not the principal component. It has to do with superpositioning and entanglement. So it's a really interesting read because they have quantum supremacy in October 2019. And now with that same Sycamore processor, now they're doing QML and basically saying, exponentially better in specific tasks over classical again.
Great. Thank you very much.
Okay, thanks. And I think we have a last question. Yeah, I hope I covered all of them with this. And did you look on migraine too? I'm asking because of the inflammatory stress response of the neurons in the brain and the possible additional chrono disruptions of the patient. This from Victoria.
Great. Thank you for the question. Actually, one of the types of disease that we are working right now is migraine. We did not a clinical trial specific for that, but we treated actually the clients of my company that are physicians, neurologists. They use it in migraine in their patients, and they found very interesting results, the decreasing of pain, but to be more robust as scientists, I would like to say that, of course, I would like to do a clinical trial or some type of more precise research style, clinical research style related to migraine. But what we have right now is case studies showing effectiveness on that, and of course safety to do that type of application of photosyntheticals. So for us, it's very interesting. And as I told you, we have approximately a few dozens of patients that we use it for migraine, and we had very good results decreasing the pain, even without knowing very precisely what was going on in a molecular or cellular level, and even without a clinical trial. So it's one of just just to point it out, it's one of the differences when we are doing research in academia or doing research in a company. Sometimes in academia, it's more focused in why it is working, why this works, and sometimes in the therapeutical companies, it is discovered the effect, nice, it works, and afterwards we go to the partnership with the academia, with the universities, to discover why it is working. And of course, when we discover, if we discover why it works, it's much easier to produce new therapeutical products to improve the previous discovered effect. So migraine, it's a big deal. It's a big market and very interesting disease to be researched. In the same way as, for example, fibromyalgia, that is a very complex disease too, and it's one of the diseases as migraine that we would like to work on that, not only inside of the company, but also doing partnership with universities around the world. So I hope I have answered appropriately.
Okay, thank you very much. And if there are not any other questions, I think we can thank you for the amazing presentation and for the amazing research that you're doing. And we'll... Oh, yeah. Oh, yeah, right. So next week, there's no presentation, but we will meet again on October 21st for chapter 5.
Yes.
Yeah.
No, I just would like to know that I'm very available person, and I put here my email, marcelo@brightmed.com.br, and please make contact with me. It will be a pleasure to talk with you all. And thank you very much for the opportunity to talk with you. It was very interesting to me. Thank you.
Thank you. Thank you for the amazing presentation. And yeah, bye everybody. And thanks for joining, and see you in 2 weeks.